Beer. It's a staple of college life and beyond. Millions of gallons of beer are consumed every day. What exactly is beer? And how can beer production keep up with ever-increasing demand?
The solution used to make beer is called wort. Wort is composed of maltose and sugar. In the fermentation process for yeast, wort is used.
Brewer et al. Conducted a study to select the strains of yeast best fit for this process. These traits include the ability to complete as many cycles as possible without affecting the final product's quality because the ever-changing conditions such as levels of O2, CO2, ethanol, and nutrients are highly stressful to the yeast. 3 species of yeast were used in this experiment. Saccharomyces cerevisiae, Saccharomyces cubanis, but the focus was on the Saccharomyces pastorianus. The original strain was A15. From that, A219 was selected to perform quicker in the early fermentation cycles where the sugar concentration is much higher and imposes hyperosmotic stress. A197 was also grown from A15 and it is an ethanol-tolerant strain. The strain can survive these stressful conditions for an extended period of time. Finally, strain A218 was bred from A197 and is both osmotolerant and ethanol-tolerant. Figure, we see the alcohol by volume percentages after fermentation done on a 2-liter scale were conducted. Both A15 and A219 were strains selected for only one trait, but yielded in an inferior product to even the original Saccharomyces pastorianus strain. In fact, A218 clearly produced the most concentrated product because it was both able to withstand the early, highly pressured cycles and also carry out repeated fermentation for a longer period of time.
What's going on in this experiment from a BIOL standpoint is the yeast strain is being selected for adaptively by its ability to both resist high ethanol concentration during the end of that 7-day process where the yeast is fermenting inside of the wort, and also to resist the heavy fluctuation on osmotic conditions when the wort or the beer production is actually finally stopped, drained out, and then a new wort solution is introduced. The reason this process is even able to happen and we can enjoy ethanol alcohol from the yeast is that ATP is being synthesized by the yeast to power its own reproduction ability. We have the osmotic conditions that it's changing. You can recall that hyperosmotic conditions are the high solution quantity inside of the cell, the low solute concentration outside the cell. So then you have water going from solution into the cell, or hyperosmotic conditions, being that there is a low solute concentration inside the cell, high solute concentration outside of the cell. So you have water transferring from the cell into the solution. This, of course, plays back into the adaptive selection as whether or not the yeast is able to withstand these heavy changes in conditions. And then finally, we have yeast reproduction. And why this is all important in the terms of yeast reproduction is yeast reproduces asexually. So the yeast's ability to both resist the high ethanol concentration and the heavy changes in its osmotic conditions is because if you have a good strain of yeast, then this will continue on for some time because there's very low variation genetic coding from one strain or one generation to the next. And this is why in this experimentation, this all began from one strain in order to test that single strain's ability to withstand these conditions. Though the hyperosmotic resistant yeast strain showed a reduced brewing time and a higher ethanol conversion efficiency than the other strains present, there was notably worse taste and reduced carbonation in the brew. Beer produced by this strain will not be able to meet market demands. So the experiment was a success in terms of making a hyperosmotic resistant strain. Further research is going to be required to help solve these lingering issues. That concludes our presentation. We hope you learned something.